Welcome to KJR SummaryCast. SummaryCast provides audio summaries of KJR articles created using an in-house AI tool with editorial or author review. For full details, please refer to the original article. Welcome to SummaryCast.
Today, we're looking at a paper that tries to bring a bit of clarity to a term that gets used a lot, especially alongside radiomics. It's titled Uncover This Tech Term, Tumor Habitat Analysis.
This was published in the Korean Journal of Radiology in 2026.
And it gets right to a core tension. For a long time, radiological imaging has worked on an implicit assumption that a tumor can be summarized as a single object.
Right. You draw a boundary around it, you measure it, you maybe extract some global features. One tumor, one entity.
Exactly. But contemporary tumor biology tells a very different story. It emphasizes profound intratumoral heterogeneity. Basically, the idea that a tumor is not a monolith. It's a complex, evolving system with different parts doing different things.
And that heterogeneity is what drives things like treatment resistance and recurrence. So the single object assumption is at odds with the biological reality.
Precisely. Now, radiomics was a step towards capturing this. The idea was that data-driven quantitative imaging features could get at these heterogeneous tumor phenotypes.
So you extract hundreds of texture features, shape features, intensity features, and use machine learning to find a representative signature.
You nailed it. But the authors point out a couple of hitches. The relationship between those radiomic features and the underlying biology often remains a black box. And in the process, you often lose critical spatial information. You know that a tumor is complex, but you don't know where.
Which is where habitat analysis comes in.
Right. Habitat analysis tries to reconcile the imaging with the biology. It conceptualizes a tumor not as an object, but as an ecosystem. An ecosystem composed of spatially distinct subregions or habitats.
Okay, so define habitat.
In this context, a habitat is a subregion of the tumor where the cells share a similar microenvironment and, presumably, similar biological behavior. Think different neighborhoods within a city.
And these neighborhoods aren't defined by anatomical boundaries, but by what? The imaging signals?
That's the key. They are defined computationally. In a nutshell, habitat analysis divides a tumor's volume into multiple subregions based on voxel-wise similarity across multiple imaging sequences.
So a habitat is a cluster of voxels that look mathematically similar on, say, T1-weighted, T2-weighted, and diffusion-weighted images combined.
Exactly. The authors make a really neat point here. They say this is essentially a computational and quantitative extension of what expert radiologists already do.
Explain that.
Well, a seasoned radiologist looks at a multi-parametric scan and mentally distinguishes different areas, right? This part looks more aggressive, this part looks like it's responding to treatment, this area seems to have low cellularity but high vascularity.
Got it. So it's formalizing that cognitive process.
Precisely. Let's get into the mechanics. How are these habitats actually computed?
I assume it starts with a lot of data.
It does. It typically begins with co-registering different image sequences from a multi-parametric MRI exam. So you'd have your contrast-enhanced T1, your T2 DWI, perfusion imaging, all lined up perfectly.
Okay, so every voxel in the tumor now has a set of corresponding values, one from each sequence.
That's the move. Each voxel is represented as a vector of these imaging values. Then an unsupervised clustering algorithm like k-means or a Gaussian mixture model is applied.
Unsupervised, meaning you don't tell it what to look for. You just say, find me 3 or 4 or 5 distinct groups of voxels based on these signal patterns.
You got it. The algorithm groups the voxels with similar signal patterns into distinct clusters, and those clusters are your habitats. But the paper flags two conceptual issues here that are often underappreciated.
What's the first one?
Consistency. This is a non-trivial problem. If you run the clustering independently for each patient, habitat 1 in patient A might be biologically totally different from habitat 1 in patient B.
So the labels become meaningless across a population. That's a red flag.
It's a huge challenge. The authors mentioned that to address this, pooled approaches have been explored. You take all the tumor voxels from a large training cohort of patients and cluster them jointly.
And that gives you a set of stable cluster centers, or centroids, that you can then apply to new patients to assign habitat labels consistently.
Right. The goal is to enforce consistency so that habitat X represents the same tissue state across all individuals. But this requires careful standardization and external validation.
Okay, what was the second issue?
The second issue is that habitats aren't so much discovered as they are imposed.
Meaning?
The analysis isn't a neutral revelation of biological truth. It's a structured abstraction that's constrained by every methodological choice you make, which imaging sequences you include, how you normalize the data, which clustering algorithm you use.
So the resulting habitat map is a model of reality, not reality itself. And the boundaries are probabilistic, not deterministic.
Exactly. The paper mentions this often involves soft assignment, where a voxel might be assigned, say, an 80% probability of belonging to habitat X and 20% to habitat Y. It adds a layer of abstraction that, again, highlights the need for rigorous biological validation against histopathology.
This brings us to what feels like the core argument of the paper. The authors seem very keen to clear up confusion between two related but different concepts.
Yes, this is a central point. The distinction between habitat imaging and habitat radiomics. They are fundamentally different constructs.
Let's break that down. What is habitat imaging?
Habitat imaging produces a spatial map. It's a visual partition of the tumor into what are presumed to be biologically different regions. Its value lies in its localization.
It's a picture, something you can actually see and point to.
Precisely, and that makes it directly actionable. Surgeons, radiation oncologists, interventional radiologists, they can act on this information. The classic example given is dose painting in radiotherapy,
Where you deliver a higher radiation dose to the habitats identified as more aggressive. That's tangible. You can see the clinical utility immediately.
You can. Now contrast that with habitat radiomics.
Okay, what's that?
Habitat radiomics takes the next step. It treats each habitat not as a region on a map, but as a new, smaller unit for feature extraction.
Wait, so you go through all the trouble of creating these habitat maps, and then you just run standard radiomics on each habitat separately?
That's the workflow. You extract texture, intensity, and shape features from within each habitat, and then use those features to build a predictive model, just like with conventional whole tumor radiomics.
So the output isn't a map anymore. It's a prediction, a number, a probability, from a model made of invisible parameters.
Exactly. And the authors argue this is where things can get very fragile.
They call it the double uncertainty problem.
Yes, the first layer of uncertainty comes from the unsupervised clustering itself. As we discussed, there's rarely a ground truth for habitat boundaries. Small variations in scanners or pre-processing can lead to different habitat maps.
So the foundation is a bit shaky. What's the second layer?
The second layer is all the well-known uncertainty that comes with radiomics modeling in general. Issues with reproducibility, generalizability, and overfitting.
And these two layers of uncertainty don't average out. They compound.
They compound. A model built this way has to first assume that the habitats it's starting with are biologically meaningful and stable. Then it has to assume that the radiomic features it extracts from those habitats are also stable, reproducible, and prognostically relevant.
In practice, that sounds like a recipe for results that are statistically impressive but clinically brittle.
That's the author's concern. They also point out that it creates a nested data structure. The habitats within a patient aren't independent observations. And maybe most importantly, it kind of defeats the purpose.
How so?
Well, habitat analysis was intended to bring back spatial interpretation, but habitat radiomics reverses that. By encoding relationships among voxels into high-dimensional features, it distances the analysis from the underlying biology and makes biological validation even more complicated.
So if habitat radiomics is so fragile, why do it? Why has this field grown, particularly in one area? The paper notes a real predominance in neuro-oncology.
It's true. habitat analysis has flourished in brain tumor imaging, especially for glioblastoma, and the authors say this is not accidental.
Why the brain?
A few converging factors. First, brain MRI offers a uniquely rich multi-parametric environment. Routine protocols already include multiple contrasts that are sensitive to different tissue properties: cellularity, edema, vascularity. It's the ideal input for clustering algorithms.
Makes sense. The raw material is already there.
Second, the biology of the tumor itself. Glioblastoma is the archetype of intratumoral heterogeneity. It's aggressive, adaptive, and spatially diverse.
It's the perfect test case for a method designed to map heterogeneity.
Exactly. And third, and this is a big one, is the clinical context. The brain presents a no biopsy dilemma.
Meaning that tissue sampling is invasive, risky, and spatially limited. A single needle biopsy from one part of the tumor might not represent the most aggressive component, which could be centimeters away.
Precisely. So in this context, habitat analysis can function as a form of virtual biopsy, offering a way to characterize the entire tumor volume non-invasively.
And that enthusiasm hasn't translated to other organs, where imaging might be less multiparametric, or where getting a tissue sample is more straightforward.
That's the argument. These converging factors in neuro-oncology create a unique niche for this technology.
So let's land this. What's the bottom line conclusion from the authors?
They frame habitat analysis as neither a panacea nor a passing fad. At its best, it's a formalization of radiological intuition, an attempt to respect the spatial complexity of disease.
But the implementation matters a lot.
Critically, the authors stress that when it gets combined with radiomics, it can become an exercise in methodological excess, divorced from biological validation and clinical actionability.
The key takeaway for me seems to be the need for clarity. Don't conflate habitat imaging with habitat radiomics.
That's it. Habitat imaging, the map, is interpretable and potentially actionable. Habitat radiomics, the predictive model, is fraught with compounded uncertainty and a lack of interpretability.
And the paper suggests that clinical value shouldn't be judged on predictive performance alone.
Right. The real tests are interpretability, robustness, and the ability to actually change patient management. A high AUC score from a black box model built on a shaky foundation doesn't help anyone make a better decision.
The final sentence seems to sum it up. Habitat analysis will only endure to the extent that it remembers this principle.
Indeed. That's all for today's episode of SummaryCast.